afternoon, everybody, and welcome to this afternoon's exciting seminar. I am super excited to be able to welcome Dr. Nicole Robb from the University of Warwick. Nicole and I worked together in Achilles Kapanides lab and a while ago, and have both spun out successful companies. There's a lot in common. I'm really grateful to her for giving you this talk on indeed some of the data that she's actually got on one of the early instruments from Exciting Instruments that went out there. I just wanted to take this opportunity because we've got quite a few new people on the call to explain a bit about who we are as Exciting Instruments and what we do. We were spun out from from my research lab at the University of Sheffield in 2021, and we make this flagship instrument is the EI Flex, which Nicole is going to talk about some of the data from that. It's an instrument for single molecule FRET and for fluorescence correlation spectroscopy. We've now got 12 customers around the world in 6 different states in the US, in the UK, and in Europe. And we're about to expand dramatically, actually. And so we're looking to hire people across all areas of the company. And so if you're interested in joining us in any way, do get in touch. The details are here on the screen. I'll put these at the end as well. We'll have a limited time for questions at the end, but we're really happy to extend that either with emails or another other calls with individuals at other points in the future. If there's something that Nicole talks about or applications that come up as you're thinking about the work that she's done, we'd be super excited to talk to you about any of your virology applications. Many of our customers work on different DNA protein interaction systems, and so some of Nicole's work will speak to that. In fact, we had the first paper published from Edwin Anthony's lab using Flex data a few months back now on a DNA protein interaction system as well. And in fact, Nicole's work that she's going to talk about today is already available on bioRxiv, so you'll get a lot more detail from that if you want to look there. So we're super excited to have you with us. The Flex is great, as Nicole will talk about it, because it really makes single molecule approaches super accessible for everybody. And so even if you're not a single molecule expert, and especially if you're not a single molecule expert, this is a piece of kit that literally sits on the benchtop, and an undergraduate can be trained in 20 minutes to get high quality publication data really, really rapidly. And so without further ado, I'll hand over to Nicole. Nicole's on the screen. Hopefully you see her there. She did her PhD in Oxford in virology, and then joined Achilles Kapanides lab, similar sort of time to me, actually, about the same sort of time there. She then rapidly progressed to a Royal Society fellowship in 2017 to establish her own lab, initially in Oxford, and then went on to gain a full lectureship position. I think they call them assistant professors actually at Warwick, don't they? Which is more internationally understood. In 2022, shortly after that, she actually spun out her own company, Pictora Bio, in 2021, where they're using some of the technology that she developed, with Achilles and others for a diagnostic point of care instrument. And so we have a number of interesting intersections in our past. And with that, I'll hand over to Nicole. I'll stop sharing my screen, and Nicole, you share yours, and we look forward to your talk. Thank you so much.
Brilliant. Thank you, Tim. Let me just get this sharing. Brilliant. Great. I hope everyone can see that and can hear me all right. Good afternoon, everybody. It's so great to be here, and wonderful to have this chance to have a chat to you guys today. I'm going to be telling you a little bit about our recent work using single molecule FRET to study viral replication complexes. So I wanted to start just by saying thank you to all of the people that actually did the work and made it all possible, which are all the members of my group at the University of Warwick. So we are a very interdisciplinary group. We combine biology, virology, and physics to study viruses. And we're really interested in how these viruses replicate and also developing new methods to treat and diagnose them. And so most of the work that I'll talk about today has been done by Danny, who's a final year PhD student in the lab, and postdoc Rory, with a bit of help from a former postdoc Andrew as well. So thank you to them. So we use single molecule methods. These are different from your traditional ensemble biochemical methods because they allow you to look at just single molecules at a time. And that's got lots and lots of advantages. So for example, being able to observe and measure single molecules allows you to uncover distributions where in a population average, you wouldn't be able to tell things apart. It allows you to study populations that don't need to be synchronized. It allows you to see very transient or rare events that would otherwise be lost in the crowd. And of course, sort of with all of the advances in modern microscopy, single molecule measurements give you the ultimate sensitivity without any amplification. So our favorite single molecule method is single molecule FRET. This is essentially the distance-dependent transfer from a donor fluorophore to an acceptor fluorophore. And you can see here that by relating the energy transfer between these two fluorophores to distance, a lot of people refer to this as a molecular ruler. And the interesting part for us is that this is a ruler that works on the length scales of around 2 to 10 nanometers. And what it gives you is information at the protein or nucleic acid level. So we can use single molecule FRET to measure dynamics and interactions between proteins, between nucleic acids, DNA or RNA, and between proteins and RNA itself. And this is just showing you that when you have the two fluorophores positioned on your favorite molecule of interest or your favorite system of interest, for example, a protein, when those two fluorophores are positioned close together, you get a high FRET signal. When they're further apart, you get a low FRET signal, and you can quantify that as well. So we do most of our single molecule measurements in solution. And to do that, we use a solution-based confocal microscope. So this is a very general schematic of how a microscope like that might work. So you often have two lasers, and here we've got a red and a green laser that alternate very rapidly. The laser light is focused often into an optical fiber, not always. And then the objective focuses that into a very, very small confocal volume. So that confocal volume is just less than a femtoliter in size. And the key principle here is that you take your labeled molecule of interest and you dilute it down so that on average only one of those molecules transits through the confocal volume at a time. You can then measure the photons emitted from your molecule of interest as it goes through that confocal volume. And this is detected on some very sensitive detectors like APDs, for example. In our case, all of this is packaged up very nicely into a flex, which, as Tim mentioned earlier, is very, very easy to use, very reliable, and has allowed us to gather some really nice data. So as I mentioned before, the key part to these single molecule FRET experiments is having a very, very focused laser beam that allows you to have a very, very small confocal volume to measure within. And with the idea that just a single molecule passes through that confocal volume at a time. And the guiding principle behind this is to maximize the signal from that one molecule that you're observing and minimize the noise of all the other molecules diffusing around it. Another really key thing that we use is alternating laser excitation, or ALEX. This was pioneered by Achilles Kapanidis in the early 2000s. And what you do here is you take your red and green lasers and you alternate them very rapidly as molecules are diffusing through the confocal volume. This allows you to extract the photon information from molecules and tell the difference between basically the light that's emitted from a donor and an acceptor fluorophore. What this allows you to do is distinguish two ratios. So we have E, which is our FRET value, which is essentially telling you the distance between the two fluorophores that you're measuring. And then we have a second ratio, which is S, or stoichiometry, which is a distance-independent measure that tells you something about the nature of the molecules that you're measuring. You can plot these two things on something called an ES histogram. So here you've got the E, the FRET efficiency, along the bottom axis, and stoichiometry along the y-axis. And one of the neat things that you can do here is you can threshold according to stoichiometry so that you can just isolate the signal that comes from double-labeled molecules. In that way, you can ignore, for example, a protein that's labeled with just a donor-only fluorophore or just an acceptor-only fluorophore, and just concentrate on the double-labeled ones, which is the ones that you're interested in. So how does it look in practice? This is some data taken from the Flex using a set of confocal standards. So what these are double-stranded DNA, where the two fluorophores are placed at very specific distances apart. In this particular standard, the fluorophores are placed 8 base pairs apart on the DNA, which is quite close together. And when we started out our experiments, we were pleased to see that this standard gave us a high FRET population, around 0.9. If you move that red fluorophore further away from the green fluorophore, so the 18 base pair standard, you get an intermediate FRET population. And if you move that even further away, you get a low FRET population. And so this is the kind of data that I'll be showing throughout the talk. And yeah, hopefully that all makes sense so far. So when we started our single molecule FRET experiments, we realized quite quickly that this is a really great tool to study the things that I like most, which is viruses. And I started off by working on the influenza virus. At that time, there were no crystal structures, there were no cryo-EM structures of the RNA polymerase, which is the viral enzyme that copies the viral genome. And we didn't know what the structure of the promoter was. So one of the big mysteries was what kind of secondary structure does the viral promoter form? And there were lots of different models put forward. There was the duplex model where the two promoter strands were basically bound tightly together. A fork model where they were separate at the 5' and 3' ends. And then a corkscrew model where the 3' and 5' ends of the promoter formed a very distinctive structure that was recognized by the RNA polymerase. So we were able to place fluorophores at particular positions on these RNA fragments and use single molecule FRET to basically clear up a mystery that had evaded the flu community for many years. We were able to show that the promoter indeed does form this very unique and distinctive corkscrew model. And this was later validated when cryo-EM structures of the flu polymerase came out. And we were also able to follow up this work by exploring many other aspects of replication and initiation in the influenza polymerase. So I've just summarized that in this schematic here. So we were able to show differences between different types of promoter and also the dynamics of what happened to the RNA as it was bound and started to be copied. But the work that I'm going to talk about today is on a different system altogether, which is on the virus that causes COVID-19. So this obviously needs very little introduction. COVID-19 has affected all of us over the last 4 years. This is just a diagram showing the number of COVID deaths reported to date. So earlier this month, we passed the 7 million mark for deaths worldwide from COVID. This is just the official reported deaths, and unofficial figures suggest that this is much, much higher, as high as 25 million people. So despite the really devastating impact of this virus on the whole world, when you have a look on the NHS website, there are actually only 4 treatments available for COVID. So we have 1 monoclonal antibody treatment and 3 small molecule inhibitors that are actually licensed for those that are at very high risk of becoming very ill or dying from COVID-19. So we thought that this was a bit of a gap, and we were interested in using our techniques to study how the COVID virus replicates and test how inhibitors work against it. So the virus behind COVID-19 is called SARS-CoV-2. It's a small envelope virus. It's around 100 nanometers in diameter. And the predominant structural protein is the membrane protein, and then there are around 24 spike proteins coming out of the virus. Inside, the genome is packaged very tightly with nucleocapsid protein, and then there's a smaller envelope protein which is embedded within the membrane protein core. As one of our side projects, we were interested in visualizing these viruses at high resolution. So we use super resolution or D-storm imaging to look at SARS-CoV-2 particles immobilized on a surface. And this is the kind of images that you get. So what we did is we used an antibody against the nucleocapsid stain, which should mark the center of the virus. And then a second antibody, which is shown in purple, against the spike protein, which are the proteins around the outside. And when you zoom in on these, you can see very beautifully that the nucleocapsid forms a dense circular structure in the middle of the virus particle, and the spike proteins are situated around the outside. We were also able to do some size estimation of large populations of these imaged particles. And we really nicely confirmed other studies that use, for example, like electron microscopy to look at the virus particles, that we got roughly around the right size for them. So if we measured the size of the signals, the particles stained with spike protein, stained for the spike protein, we got around 94 nanometers on average. If we looked at the nucleocapsid, this was slightly smaller, so representing the inside of the particle, and the envelope was sort of in the middle. So this is a really nice validation of the size of particles. For today's purpose, though, what we're interested in, or the part of the virus that we're interested in, is the RNA polymerase. So SARS-CoV-2 has a single-stranded positive sense genome. It's a really long genome, it's around 30 kb in size. And this is replicated by a very special enzyme called the RNA-dependent RNA polymerase. This is a viral enzyme, so it's made by the virus when the virus enters the host cell. And it's a large complex, but for the purpose of our studies, we use a minimal version of the complex, which is made up of 3 non-structural proteins. So these are NSP12, NSP7, and 2 copies of NSP8. There are several cryo-EM structures of the RNA polymerase available, either with or without RNA present. And from these structures, they're very, very useful. They allow us and guide us on where to put fluorophores for our studies. And they've also given a lot of information about the different functions that the different non-structural proteins have. So we know from these studies that NSP12 is the main polymerase. It contains the catalytic domain, or the active site, that actually copies the RNA. It also has capping function. The 2 NSP8 proteins, which are shown here in purple, have primase activity, and they're also thought to help processivity of the enzyme as it moves along the RNA. So you can see there's these 2 long, they're called sliding poles in purple, and these contact the RNA as the polymerase moves along it when it's copying. And then there's a small cofactor, a small protein called NSP7, which is shown in dark blue. And this, the function isn't known, although it has been suggested that this is what holds the whole complex together. So why do we care about RNA polymerases? Because these are a really important antiviral target. Out of those 4 available licensed treatments that we have for COVID-19, 3 of them are targeting the polymerase. So it's a really important therapeutic target, and that's because it plays obviously a pivotal role in viral replication. Without it, the virus can't replicate or cause disease. It's also virally encoded, so the host, as in humans, they lack a functional equivalent. And that means that if you target this polymerase, there shouldn't be any target-related side effects to the host. The aims of our study were to see if we can reconstitute an active minimal replication complex in vitro, to see whether we could analyze and observe RNA synthesis by this minimal complex using single molecule FRET. We wanted to validate whether single molecule FRET was actually a reliable technique for investigating this. And finally, we wanted to investigate the mechanisms of action of key inhibitors on the SARS-CoV-2 polymerase at the single molecule level. So we started by purifying a minimal replication complex. To do this, we had to express and purify the 3 protein subunits of the polymerase separately. So we expressed the NSP12 protein in a baculovirus system and NSP7 and NSP8 in insect cells. We purified them, and as you can see here, when we ran them out from a gel, we got nice pure bands of the expected sizes for all of the different NSPs. As well as having a wild type NSP12, we also expressed a mutant version of NSP12, which has 2 point mutations at positions 760 and 761. These are, we know from structural studies, these are the amino acids that are involved in coordinating the metal ions needed for catalysis. So by mutating them, it's essentially an active site mutant. So having successfully expressed and purified the individual components of the replication complex, we next wanted to test whether we could actually get any in vitro activity with them. So we started by adding the different components together. So we reconstituted first the polymerase by adding in the correct ratios of NSP7, 8, and 12, and then mixing this with fluorescently labeled RNA template and primer strands. So this is what the RNA looks like in a bit more detail. So we have a long template strand shown in orange and a short primer strand shown in blue. The template strand has got a green Cy3 dye on it, and the primer strand has a red Cy5 dye. And when you add polymerase and nucleotides to the system, what we expect to happen is the polymerase binds and is able to extend the primer from a 20-mer to a 30-mer. And because that primer is labeled with a red fluorophore, that means that we should be able to see a red labeled 20-mer extended to a red labeled 30-mer. And this is exactly what we observed, which we're really excited about. So on this gel, what you can see in red at the top is the position of the primer, and in green is the position of the 20-mer of the template. And then here, what happens when you add wild type polymerase as well as nucleotides is you get extension of that 20-mer primer to a 30-mer. And we also have all of the relevant controls, and we tested what happened when we swapped the red and green dyes around. And we saw that basically the nature of the dye didn't make any difference. So if you have a green dye on the primer, this will also be extended to a 30-mer when you add nucleotides and polymerase. And this is just a quantification of the gel showing that we very nicely get activity of our in vitro system. So next we wanted to use the system in a single molecule FRET extension assay. So if we use the RNA design that I showed just a couple of slides ago, you basically wouldn't get any change in FRET when the RNA was being copied, because the 2 dyes would move together and the polymerase would move in the opposite direction, and there wouldn't be an observable change. So to do an extension assay, we changed the structure and the sequence of the RNA slightly. So we kept our primer the same, although we moved the red label to position 12 rather than position 1. But our template we redesigned so that it had a self-annealing hairpin loop in it. And it still had the green Cy3 dye on it as well. So when you anneal these two RNAs together, you basically get a hairpin RNA that looks like this. And what we expected is that the two dyes would be very, very close together, so you would get a high FRET signature. If you add polymerase to the system, and as well as giving it some NTPs, what we expect to happen is that the polymerase would start copying the RNA, it would start extending the primer until it ran out of NTPs, and it would open up that hairpin at the end. The green dye would therefore move further away from the red dye, and you would get a drop in the FRET to low FRET. One of the neat things about this is that you can control the extent of extension that you get. So if you add just ATP only, you would get extension of the primer from a 20 mer to a 34 mer, and then it wouldn't be able to extend anymore because it doesn't have any UTP present. And if you add both ATP and UTP, then the polymerase is able to extend the primer all the way to the end of the template strand. So it would extend from a 20 mer to a 43 mer. We also did some accessible volume modeling of the fluorophore positions in our just as a sanity check to see whether we would indeed get a change in FRET as we expected. So here we've modeled the RNA in the hairpin conformation, and you can see that the two dyes, as expected, would be very close together and give us a high FRET signature. And then if we have full extension and we model the RNA as being fully extended, those two dyes move much further apart and we get a very low FRET signature. We also did check this on a gel beforehand. And so what you can see here is, again, in red, the position of the primer, and in green, the position of the template. When you add polymerase and either ATP only or both ATP and UTP, you get really nice extension to either a 34 mer for the ATP only or a 43 mer for the ATP and UTP, just as we would expect from the design of our RNA. And again, this is just the quantification of the gel. So we did all the checks and everything looked really good. So the next thing to do was to actually test this in a single molecule FRET experiment. We started by testing the RNA only. So this is the RNA that's been annealed to form the self-annealed hairpin. And just as we expected, we get a really nice high FRET population centered at around 0.9. As a control for what extension should look like, we annealed two fully double-stranded RNAs together, which mimics what we would expect if the RNA polymerase and nucleotides were added and the primer was fully extended. And indeed, as expected, we get a nice shift to low FRET with that double-stranded RNA construct as well. We then went back to our self-annealed hairpin. We added the polymerase and we added ATP. And we were really pleased to see that, as expected, we get some partial extension of the RNA. So here we have two FRET populations. The high FRET population is non-extended hairpin RNA, and the low FRET population is the partially extended RNA, where the polymerase has managed to add ATP, but then not managed to finish fully extending because there's no UTP present. When we add the same thing, but with UTP as well, you get almost full conversion to the fully extended primer, which is just what we expected to see, and we're really excited to see that we could actually observe extension with our single molecule assay. As a control, we used our mutant polymerase, which was the active site mutant that wasn't able to extend RNA. And indeed, you get almost no extension there, and the RNA just remains in its hairpin-bound form. We also did some controls just to validate that what we're seeing was correct. So we tried to move the dyes around to different positions. And when we moved the red dye from position 12 on the primer to further in to position 19, which is where we actually expect the polymerase to bind, so it's almost in the active site itself, we didn't get any activity, as you would expect. And when we moved the green donor dye, when we moved it from the end position, the 5' end of the template, further into position 3, we get really nice conversion from the hairpin structure to a fully extended structure, just as we would expect. So we were really happy that we had an assay that appeared to show RNA extension at the single molecule level. We had an in vitro system that replicated really nicely. And as a kind of final control, so what we noticed, if I go back here, what we noticed is that when you had ATP only rather than ATP and UTP, you get this partial extension. So you get still some hairpin RNA there, as well as some partially opened RNA. And we hypothesized that what was actually going on is that when you add just ATP, the polymerase is adding the ATP and then it's stalling because it can't continue. Whereas if you add the ATP and UTP, basically the polymerase copies all the way through to the end and then falls off. And we were able to confirm this in this native gel here. So this is a gel that allows you to observe when things are bound together or stay complex in the gel. You can see in the second lane that when you have fully extended RNA, you get that bright yellow band there. And then when you add polymerase to this, but without any NTPs present, you don't get that band formed, but you do get a band much higher up in the gel, which represents polymerase bound to RNA. So it's shifted upwards in the gel. If you add just ATP, that high band gets much stronger, suggesting that the polymerase is stalling and remains bound to the RNA. And if you add ATP and UTP, we expect the polymerase to extend the primer fully and then run off the end. That band then decreases. So this confirmed our hypothesis that if you add limiting NTPs, that stalls the polymerase on the RNA. So that's the double-stranded band that you would expect when you get full extension. And these are the bands that correspond to polymerase-bound RNA in its different forms. So having validated our assay and we're really pleased that we could see extension, we next wanted to move on and see if we could use it as a screen to see how inhibitors work against the viral polymerase. We started off by investigating the mechanism of action of remdesivir triphosphate, which is one of the only licensed drugs that we have for COVID-19 at the moment. Remdesivir is a ATP analog, and its proposed method of action is delayed chain termination. So what do I mean by this? In our extension assay, what's hypothesized to happen is that if you have the RNA only, you have the hairpin form and you have low FRET. Whereas if you just add polymerase and normal NTPs, that will then convert to a low FRET signature as the RNA opens, as it's extended. But if you add remdesivir, what you would expect to happen is instead of the ATP, remdesivir would be incorporated. The polymerase would stall when remdesivir is incorporated, and basically not be able to finish extension. The delayed part refers to the fact that structural studies suggest that when remdesivir is incorporated, the polymerase is able to incorporate another 3 nucleotides before it stalls and falls off. That's because there's a structural clash or steric clash between a part of the polymerase and the RNA. So this is what we expected to happen, having read the literature on it. And so what we tried to do is basically add remdesivir into our experiments and see what happened. So we started off by running one of our fluorescent gels, where we basically, what you see along the bottom is the position of the primer RNA, and the top band is the position of the extended primer. If you add just the polymerase alone, you don't get any extension. If you add polymerase and ATP, you do get a small amount of extension, so you get a slightly smaller product. And if you add normal ATP and UTP, where you'd expect full extension to happen, as well as increasing concentrations of remdesivir, we were actually quite surprised with what we saw in the gel. So what we expected is that with increasing concentrations of remdesivir, we would get reduced amount of the full-length 43-mer product formed. But actually what we could see is that the more remdesivir added, actually the higher this 43-mer band was, or the more intense this band was, suggesting that remdesivir doesn't stop full extension of the RNA, and in fact might even be misincorporated. So the concentration of the natural ATP and UTP we were using here was 3 millimolar, which is quite high. So we repeated the experiment with much lower concentrations of the natural nucleotides. So here we use 0.3 millimolar of ATP and UTP under the same conditions. And this result was much closer to what we were expecting, in that as you increase the concentration of remdesivir, you get a decrease in the fully extended product. And at 5 millimolar remdesivir, you can see quite nicely there's that ladder effect on the gel. So you are in fact getting chain termination. However, in order to see this effect, we needed to add very low natural NTP conditions. So how did this look in our single molecule FRET assay? So here we've got 3 millimolar ATP and UTP, which is the high NTP conditions. In the top, we've got just no remdesivir present, just the polymerase and ATP, UTP. And as expected, we get a really good conversion of the hairpin into the low FRET extended product. As you increase the concentration of remdesivir, you essentially get a higher proportion of the extended product, which closely matches what we'd seen on the gel. Next, we tried the same assay with our low FRET, with our low NTP conditions, expecting to see that we would basically retain the hairpin conformation of the RNA and not get much extension. But to our surprise, we actually saw very similar results to the high NTP conditions. And I'll explain why this is over the next couple of slides. As a control, we also did the same experiment with our mutant polymerase, which is the active site mutant. And this nicely confirmed that if the polymerase isn't able to extend the RNA, we don't get any conversion from the high FRET to the low FRET population. So in order to explore a bit further what was going on in the low NTP conditions, so just to remind you, our gel suggested that you do get chain termination by remdesivir in low NTP conditions. However, our FRET results suggested that the RNA wasn't in the hairpin conformation, was still able to extend outwards and form a low FRET population. So again, we turn to our native gel electrophoresis, which is where you can see complexes bound together on the gel. The schematic at the top is showing the different RNA populations that we might expect to see on the gel. So you can either observe a green labeled RNA only or a red labeled RNA only, or the two complex together. Or when you add polymerase, that complex would then be shifted on the gel. And when you add nucleotides, you might expect to see varying different populations of bound products. This is the gel itself. So at first glance, it looks quite confusing. There's quite a lot of bands there. So I tried to explain down the side what all the different bands represent. So if we do controls where we have just the fully extended product, you can see that they're marked with a little star. And when you add polymerase, like in the previous gel I showed, you basically get a big shift of the gel upwards into the gel, higher up in the gel, which represents the polymerase bound to the RNA. And then the really interesting part that we noticed was that when you add increasing concentrations of remdesivir, you also get these intermediate products, which represent stored products where the polymerase is bound to the RNA because it's incorporated remdesivir and it's got stuck, and it's stuck there and it's not able to extend any further. And the key thing that this suggested to us is that in low NTP conditions, so 0.3 millimolar ACP and UTP, we get a much more significant degree of stalling of the polymerase on the RNA than in high NTP conditions. So we wanted to confirm this using a slightly different technique. So we used the FLEX for doing some fluorescence correlation spectroscopy or FCS. So this is a technique that measures the interactions or dynamics of biomolecules by analyzing their fluctuations in fluorescence intensity as they diffuse through the confocal volume. And these are the results that we got. So we ran our normal assay under our normal conditions, which is high ATP and UTP, either with or without remdesivir present. The green line is our rhodamine 6G control. The pink line is RNA only. And then you can see that whenever polymerase is added in the presence of high NTPs, we do get a shift to the right, which is telling us that the polymerase is actively on the RNA, it's copying the RNA. But we don't see really any differences between when remdesivir is present or not. So in comparison, when you have low NTP conditions, again, the same experimental setup, where the pink line is the RNA only, but this time when we added polymerase and NTPs and either remdesivir or not, we see that the dark blue line is much further shifted to the right, which suggests that remdesivir is stalling the polymerase on the RNA to a much greater extent when low natural NTPs are present. So we put all of this together to come up with a model of how remdesivir is acting. So in the presence of low NTPs, when remdesivir is the much more likely to be incorporated into the RNA product, we get exactly what you would expect from reading the literature. You get delayed chain termination. So here, the polymerase is represented by the blue circle, and you can see that remdesivir would essentially compete with the normal ATP to be incorporated. Once remdesivir is incorporated, and the likelihood of that happening is quite high because obviously the natural NTP concentration is quite low. The polymerase stalls on the RNA and it terminates. It's not able to extend any further. The interesting thing that happens, though, is when there are high NTPs or high natural NTPs present as well as remdesivir. And this is interesting because actually this is much more representative of the situation in a cell where you expect high natural NTPs to be present. So could be representative of what happens in vivo. So here, what we could see from our gel and our FRET experiments is that the inhibition by remdesivir is overcome by the high natural NTP concentration. And in fact, remdesivir seems to be misincorporated into the RNA, and the polymerase just carries on after that. So there was two very nice studies in recent years which suggested an alternative mechanism for remdesivir action, which is that once remdesivir is incorporated into the viral genome or the viral RNA, basically the polymerase has difficulty incorporating a natural UTP opposite the remdesivir. So this suggests that there might, in fact, be another mechanism of remdesivir action in vivo compared to in vitro. Now, our results do seem to support that. Next, we wanted to test whether our assay could also work as a screen for a non-nucleoside analog inhibitor. So for this, we used a compound called suramin. This is a very, very old drug. It was described more than 100 years ago. It's a non-nucleoside analog, and it has been shown to inhibit SARS-CoV-2 in cell culture by two different methods. So it's been suggested to prevent binding of the virus into cells, but also by acting as a direct inhibitor of the viral polymerase by binding at two different sites on the protein. So there is a cryo-EM structure of the SARS-CoV-2 polymerase showing that two molecules of suramin actually bind within the active site, and they prevent binding of the RNA template strand and entry of the product RNA into the active site. So we're really interested to see how this would behave in our in vitro extension assay. What we expected was that, as usual, our RNA only would provide a low FRET, a high FRET signature because the two were close together. And when you add polymerase and ACP and UTP, so then open up the RNA and convert it to a low FRET signature. And by adding suramin into the assay, this would prevent polymerase binding to the RNA, and the RNA would stay in its hairpin structure. We tested this on a gel beforehand, as usual, and we were pleased to see that things seem to be happening exactly as you might expect. So as you increase the concentration of suramin, you get a decrease in the extended RNA product, the 43-mer. And again, if we look at this on a native gel, you get a decrease in the double-stranded, fully extended product as suramin concentration increases. We were really pleased to see then in our single molecule FRET assay that our results closely mimicked what we saw in the gel, and that suramin, as you increase concentrations of suramin, it inhibited the activity of the polymerase. So it inhibited its ability to extend the RNA construct in a concentration-dependent manner. So the next thing that I'm going to describe is just some further work that we did on the system in order to confirm that remdesivir's mode of action was different depending on the availability of natural nucleotides around. So for this, we turned to a slightly different mechanism or slightly different mode of microscopy to measure single molecules called TIRF. So here, instead of observing molecules diffusing in solution, they are immobilized on the surface of the glass slide. In TIRF microscopy, instead of directing the laser beam directly through the sample like you do in epifluorescence, the incident beam is focused beyond the critical angle so that you get total internal reflection, which means that rather than the light going through the sample, it just forms an exponentially decaying evanescent wave. And this allows you to massively reduce the background that you see in your sample as you get excitation just within a very, very small distance of the glass surface. So this within a couple of 100 nanometers, which is perfect for observing things like proteins or small viruses. So using this technique, we wanted to actually directly visualize the polymerase itself. And to do so, we had a good look at the crystal structure, the cryo-EM structure, and we decided that we would try to fluorescently label NSP7 So if you remember, NSP7 is a small cofactor that holds the complex together. It has 3 naturally occurring cysteine residues in it. So we set about mutating pairs of these so that just a single cysteine residue was retained. So we made 3 versions of NSP7, one which just retained the first cysteine, which was at position 8, which we call cysteine 1, one which just retained the second cysteine at position 32, which we call cysteine 2, and one which just retained the third cysteine position. And we expressed and purified these 3 different mutants. We ran the mouse in a gel, and here you can see the wild type NSP7 at the expected size on the gel, as well as our 3 different NSP7 mutants. We were then able to use malamide labeling to specifically label the cysteine in each NSP7 protein that we purified. So here we've taken one of them. I think this is the cysteine 2 example, and we've labeled the cysteine with Cy3, which is a green dye, and you can see those 2 bands on the gel there. So the top band is the labeled NSP7, and you can see that, and the lower band is free dye that's not incorporated into the protein. And as you do increasing washes, that free dye decreases until you're left just with a really nice population of labeled NSP7 protein. We then incorporated this into our minimal replication complex and did activity assays to confirm that mutating the cysteines in NSP7 didn't affect the activity of the polymerase. And you can see here that for all 3 mutants that we've made, we still get substantial in vitro activity when the other 2 cysteines are labeled, are mutated in each case. And when the 3 different protein preps were labeled, in this case they were labeled with a red dye, the activity is also still retained. So we were confident that we had specifically labeled NSP7 within the RNA polymerase complex, and that the RNA polymerase complex was still able to replicate and extend RNA in our in vitro systems. We then immobilized these complexes on the surface of a pegylated glass slide. So we did this either by isolating the RNA directly and pulling that down onto the surface, or using an histidine antibody that bound to a hist tag on NSP12 in order to bring the labeled complexes down onto the surface. And in this experiment, what we've done is we've labeled NSP7 with a green Cy3 dye, and we have our normal RNA, which is labeled just with a single Cy5 or red dye. And we're looking for co-localization between the red and green signals, which would indicate that we have polymerase on the surface and the polymerase is able to bind to the RNA. When we quantified this, we were really pleased to see that, in agreement with our previous FCS and our diffusing single molecule FRET experiments, when you have low NTPs present, and as well as remdesivir, the amount of co-localization, so the amount of polymerase stored on the RNA, was greater than when you have high NTPs present. So all of this was just added support to our proposed mechanism of action of remdesivir. I think that just leaves me to summarize everything that I've covered today. So we were able to successfully purify and express minimal replication complexes. We were able to show that these minimal replication complexes were active using fluorescent templates and lots of alternative labeling positions. We could show using single molecule FRET in our extension assay that we can measure FRET changes during extension. We were able to validate and verify the different mechanisms of actions of both a nucleoside inhibitor and a non-nucleoside analog inhibitor of SARS-CoV-2. And we were able to successfully label the NSP subunit of the RNA polymerase, and used TIRF assays to observe the immobilized complexes. So that just leaves me to say thank you so much to everybody involved in the work, especially Danny and Rory, who've done the bulk of the work I've shown here today, to our collaborators at the University of Warwick and the University of Oxford, and thank you to all of our funding sources as well. And thank you so much for everyone for listening.
Awesome. Thanks so much for that, Nicole. Absolutely brilliant talk there. And open the floor to questions if anyone wants to raise their hand, and then we can take them in turn if people have got any questions they'd like to ask at this point. I'll also just share my screen again with the contact details for if you've got any questions you'd like to ask after the chat, either of us Exciting Instruments in terms of the capabilities of the instrument. If you'd like to do some of these similar sorts of experiments that that Nicole's been doing, and Nicole, fabulous to see both the smFRET functionality, the FCS functionality used in good measure there. And I was intrigued that at one point you said there was an unexpected FRET result. Was that the trigger for you to then go and do those extra annoying gels, or how talk us through that process a little bit?
Yeah. Yes. So when we started, we expected it to be a nice, neat story where if you add remdesivir, it terminates and you don't get any extension. And what we saw was actually the opposite. And so we added increasing amounts of remdesivir, and the RNA was, at least in our single molecular assays, still appeared to be fully open, as though it's been fully extended. So that triggered us to test different NCP concentrations and really dig into what was going on with remdesivir. And yeah, I think the nice thing that we were able to show is that the commonly described mode of action is probably only applicable to in vitro situations. And in a cell where there's actually high NTPs present, it's much more likely that you're not getting chain termination, but you're getting some other form of template-mediated inhibition happening there.
So that's really interesting. So that actually changes the way we understand the mode of action of that particular drug.
Yeah.
Brilliant. And then obviously there's some other really nice things with was there 100-year-old remedy that you pulled out there, but which then seemed to behave as expected, is that right?
Yes, in a much more straightforward way. So this is a 100-year-old drug that traditionally is used as to treat parasites, and somebody, I guess, during the pandemic decided just to try it out on SARS-CoV-2 and was able to show that it does actually bind the SARS-CoV-2 polymerase. So this one, I think, could be really promising because it seems to have a very straightforward mode of action that's relatively easy to understand. So yeah, there could be potential there to use that one.
Fantastic. Well, we just for the interest of time, it is 6 o'clock. We're well, in the UK anyway. It's probably very different in other parts of the world, one would imagine. But it's absolutely brilliant. Thank you, Nicole, again for this opportunity. I'd encourage anyone with any further questions, either on the instrument or on the science, to email either ourselves info@excitinginstruments.com or Nicole directly. I'm sure she'll be really happy to take those questions, and also to talk about the we do have we've got time for one there. Can you see it in the chat, or shall we read it? Shall I read it out for you?
If you wouldn't mind reading it out, that'd be great.
Fine. It's from is it from Eric Holstrom? Eric says, I'm surprised your polymerase recognizes your hairpin substrate since it appears fully base paired. Similarly, did you ever look at the RdRp in the absence of any nucleotides? Is the hairpin open and closing rapidly, and does the RdRp bind to the template and trap it in a non-hairpin conformation before extending the primer?
Yeah. So it does look fully double-stranded, but there is obviously a nick there is a gap between where the primer is bound and where the self-annealing hairpin is. So I was also unsure when we tried it whether it would work or not. but it does seem to be able to just bind that very small nick in the RNA and extend from there quite efficiently. And we did also try other templates where we've increased the size of that gap, and we've shown that it does work in a similar way. So I agree, it is quite surprising. And the polymerase does seem perfectly capable of just recognizing a nick and extending from there. And yeah, to answer the second part of the question, if you have that and you just have no NTPs, it is capable of opening it and just stalling on the RNA then without being able to extend it as well.
Brilliant. That's excellent. With that, we'll just say thank you again to Nicole. It's been a pleasure to have you all here. There'll be another exciting seminar in about a month's time. We'll email everybody for that. Do sign up for our email list again at Exciting Instruments, info Exciting Instruments if you're not already signed up. And also, as I say, we will be recruiting and getting some adverts out in the coming weeks, specifically looking for application specialists. So if you know how to develop these assays in the way that Nicole's been doing, we're super excited and keen to talk to you guys about coming and joining us as we continue to expand and bring these really powerful methods to as many people as we possibly can around the world. So thank you, Nicole, again for your time today. Absolutely brilliant. Thank you all for coming, and we'll see you at the next one soon. Bye, guys.